Good afternoon, all. Welcome. And as the scientific coordinator of the Center for Philosophy of Science of the University of Lisbon, it is my great pleasure to open this session, the second session of this year, of our permanent seminar of the Center for Philosophy of Science. Today's speaker will be Constança Carvalho. Constança did her PhD at the CFCUL, and she defended very successfully the PhD last year. She is a member of our research group on philosophy of natural science, before as a PhD student, and now as a full member of the group, research group 2. And I just want to thank Constanza for having accepted our invitation to this session, and it's a great pleasure to have you here. And Sylvia, the floor is yours.
Yeah, thank you, John. Welcome, everybody. Welcome, Costanza, and thank you very much for accepting our invitation. Constanza will talk for 45 minutes, more or less, and then we will have time for the questions and answers. I will moderate the Q&A. So if you want to ask a question, just write your name in the chat, or you can also raise your hand using the reactions button here on Zoom. We are recording the session, but we will record only the part of the presentation. So during the Q&A, we will not be recording, so feel at ease in asking all your questions. Constanza, so I think I said everything I had to say. Constanza, the screen is yours. Thank you very much for being here.
Okay, thank you very much for inviting me. It's a pleasure to be here and talk about these things that are so important to me, and I guess for most of us. It's a pleasure to be here with these many people attending. So I'm going to talk to you about animal use in mental disorders research and discuss whether it is or not a necessary evil. As Professor João already mentioned, I did my PhD here on this topic, so I'm going to share with you some of the data of my PhD and some new data that is yet unpublished but submitted. Okay, so what is animal experimentation? There are many definitions, but animal experimentation can be defined as the use of non-human animals in scientific experiments that seek to control variables that affect the behavior of animals or their biological systems. It's a very old practice, at least 2,500 years old, but it's been increasing exponentially since the second half of 19th century. And the latest estimations, it's that currently at least 192 million animals are used annually worldwide for animal experimentation. Animal experimentation is conducted in many fields. One of them, one big portion, is the biomedical research, which is the research aiming to understand and ultimately cure human disorders. Here you can see the current paradigm of current biomedical research. You have an idea, a drug or a chemical, then it's tested in a simple model like a computer or an in vitro test. Then it moves to more complex models like animal models. Then it moves to to tests with humans. And then if the idea worked or the drug works, the idea is proved correct, the drug is effective, or the chemical is safe. Animal experimentation has always been a very controversial topic. For many years, there was an ethical discussion around animal experimentation. And while some claim that even though animal experimentation harms animals, it's the only way to advance medical progress, others claimed that animals are not ours to be used regardless of the benefits for humankind. But in the past few decades, there was a shift in this discussion into more an utility discussion, which are in fact animal models contributing to current knowledge of biomedical research. And this happened because there was, since late 90s and the early new century, some studies that showed that animal experimentation many times ran simultaneously to human to human trials, not experimentation, to human trials rather than sequentially. The results were different. And there was a big report from FDA in America in 2004 that was on drug discovery. And it stated that only 8% of the drugs that ever pass preclinical stages reach successfully past clinical trials. This means that 92% of the drugs that are ever tested, when they go to the humans, they don't work. And so this raised a new topic of discussion, which is if there is a knowledge transfer, because if the animal model studies are relevant, then we expect the knowledge transfer from one area to the other, to another, and if the results are the same. And my PhD and the data I'm going to share with you today follows under this umbrella of the utility discussion. And more precisely, I'm going to talk to you about the contribution of animal models to mental disorders. And why is this particularly important? Because of the prevalence level of morbidity and economic burden of mental disorders, which is very substantial worldwide. Because of the complexity of human disorders, which have human-specific symptoms that are impossible to mimic in animal models. Because the research of nervous system and mental disorders has the highest amount of severe procedures in Europe. And here I would just like to make a parenthesis to explain that under the current European legislation, there are 3 kinds of procedures. They can be considered mild, moderate, or severe. And the severe procedures, this is regarding animal experimentation, and the severe procedures are the ones who cause more pain or more lasting pain. And so they should be avoided. However, in this kind of research, many severe procedures are used without empirical data on their real contribution for human disorders. So it's very crucial to assess this. Also, the lack of recognition of animal models by clinicians who work within mental disorder fields, namely psychologists. There are some interesting surveys on this. And of course, because this has barely been done. And in science, whatever it was not been done is interesting. And there was a PhD on this topic, on the contribution of animal models to eating disorders in 1998 by Kenneth Shapiro, and that was it. And before my thesis, that was all that we had on mental disorders. So now I'm going to start introducing you to my data, to my empirical data, and this is the first paper I published on this topic. It's called The Contribution of Animal Models to Contemporary Understanding of Attention Deficit Hyperactivity Disorder. And here is the part where I will ask Sylvia not to record, because I'm going to explain to you this disorder and show an actual patient. ADHD research. Some of them are genetically modified animals, actually most of them. And these animal models, they mimic these traits: locomotor hyperactivity, attention deficit, and impulsive behavior. These 3 are traits of ADHD, but it's important to mention that in order for a patient to be diagnosed with ADHD, he needs to fulfill at least 5 of a list of 9 symptoms of attention deficit, and at least 6 out of a list of 9 items of hyperactivity. And here we have 3 traits, which is not enough. And this is very interesting, these animals, they also present memory deficit, which is not a trait of attention deficit hyperactivity disorder. These kids do good on memory tests, on long-term and short-term memory tests. So this is a bit awkward that this is considered a trait in the animal models when it is not a trait in the human beings. And in here, what I did was I conducted a citation analysis on the 211 papers published prior to 2010, resorting to animal models to study ADHD. A citation analysis consists in locating papers that follow some under some category. Here, I located in PubMed all the published papers that resorted to animal models and presented the original studies. And then I resorted to the tracking facility within Web of Science to verify how many citations these papers received and by what sort of papers. And this is a citation analysis in brief. You locate the papers you want, and then you use the citation tracking facilities to verify how many citations or what sort of papers cited your sample. Then we conducted a systematic analysis of the results. And here, 2 independent raters read the 220 and 244 human medical papers that cited the animal papers on ADHD and determined the relevance of the citation for the citing paper. Because we all know when we are writing papers, we cite many papers, and some are relevant for are crucial for our hypothesis or for our method, and some provide minor information of minor relevance, which is information that is not directly related to the question of our paper, and some are redundant because they are just examples in the middle of other examples. And here are the results. And as you can see, 43% of the papers were never cited, of the papers describing animal models for ADHD knowledge were never cited in human medical papers. The vast majority of citations came from subsequent animal studies, followed by review papers, and only a small proportion of the citations came from human medical studies. We then read the human medical papers were available for us, and the 2 independent raters verified that only 10 citations, which actually belonged to 8 papers, were relevant for the hypothesis, and none was relevant for the methods. It's important to state that out of these 10 citations, they belong to 8 papers, 3 of which describe data of humans and rhesus monkeys. And the part that was cited in the human medical paper was the human part. So in fact, only 5 papers describing animal data made a significant contribution for subsequent medical papers within this research field. This is not very good. This shows that the contribution was poor. But what about other disorders? Could this be a problem just of ADHD? So now I'm going to present to you data on major depressive disorder. Major depressive disorder is the most severe depression type. It's a very serious disorder that affects 8 to 12% people worldwide, and is one of the leading causes of disability worldwide. Why is that? Because people with major depressive disorder, they have a major depressive episode that can go from 2 weeks to several months, that sometimes they cannot even get out of bed. So you can imagine how disabling this is. And again, for a patient to be diagnosed with major depressive disorder, the patient needs to fulfill at least 6 out of a list of 9 criteria that include things like suicidal thoughts, feelings of excessive guilt, loss of pleasure and interest in daily activities, loss of interest in daily life, feelings of self-worthlessness, also changes in sleeping patterns, in eating patterns, in the locomotor patterns. And so as you can see, while in animals you can study things like changes in sleeping patterns, you cannot assess suicidal thoughts or or excessive feelings of guilt, for example. And here I did not look into all the papers that were ever studied resorting to animal models, because there were too many. But I analyzed all the papers that used rats, because rodents are the vast majority of animals that are used for biomedical research within this field are rodents. And for major depressive disorder, at the time I conducted my research, it was rats. So I conducted a citation analysis on the 197 papers that I found that described data with rats aiming to study major depressive disorder, and also the 90 studies I found using non-human primates. In this case, marmosets or rhesus monkeys are the most common models. And why this species? As I explained, rats because they are the more common, and primates for two reasons. Because primates, except in China, in the Western societies, the research resorting to non-human primates is very restricted. So, and should only be conducted when the benefits expected are very, very substantial, are very high. So it's important to, I think it's interesting to verify what was the contribution of primate models to answer for the current knowledge of major depressive disorder. And so I'm going to start by showing you the paper on rats, which was published last year, the contribution of rat studies to current knowledge of major depressive disorder, results from a citation analysis. And again, we used Web of Science and Scopus to verify the number of citations that the papers received. And then we read the abstracts and sometimes the full papers to determine what sort of paper it was. And here are the results. And once again, the vast majority of citations that these papers received were from subsequent animal research papers, and only a small proportion of the citations came from human medical studies. And 49% of these papers were never cited in human medical papers of major depressive disorder at all. So it's not looking very good again. And here it's important to state that we did not conduct a qualitative analysis of citations. So it is reasonable to assume that if we conducted that, and the percentages of papers that would have been relevant for the understanding of this disorder would be even less. Amongst several procedures that are used to study depression in rats is the forced swim test that you can see here. Forced swim test is a test that was developed in 1970s, and it consists in putting a rat or a mice in a bowl of water, and when the rat tries to stop swimming or stops trying to escape, it's considered that the rat is depressed. This is a very popular test to study antidepressant drugs and traits of depression. It's also a very severe procedure. It is described in the legislation. They describe examples of what is severe procedures. And one of the procedures that is described there is the forced swim test using exhaustion as the endpoint. This is a popular test amongst studies from depression. And a bit after the COVID took over all the research interests, there was a big discussion amongst the scientific community if this test should be banned or not due to its cruelty and lack of results. And so I decided that it would be interesting to use my data to contribute to this discussion. And so I went to my databases on the citation analysis of the 178 papers published resorting to rats to study MDD, and they verified which used the forced swim test and which used other procedures. And I verified that 43 papers resorted to forced swim test and 135 to other procedures. And so then me and my co-authors, we decided to compare the number of citations received by the papers that used forced swim tests and the others who did not use forced swim test. And as you can see, the median is not very good, but the median is even more impressive. The median citation that papers that used forced swim test received was 0. And this is a very big number, and this paper is currently submitted for publication. And in this paper, we suggest a ban of this paper, as been suggested by other authors, since it should only be used when expected benefits are significant, and because we find that in the main aim for its use, the median citations are 0, we advocate for a ban of this test. So far, we've seen that animal models have contributed very little for mental disorder research, but what about other approaches? Could this be a problem of all indirect approaches and not just animal models? This was a question I was asked by a colleague at University in Faculty of Sciences, and I thought this was a very good question. And so I decided to address this question. And the results I'm going to show you now were published in ATLA, and this paper is called The Relevance of In Silico, In Vitro and Non-Human Primate-Based Approaches to Clinical Research on Major Depressive Disorder. And as you can see for the title here, what we did was we conducted, again, a citation analysis and a systematic qualitative analysis of citation in papers that described in vitro, in silico, or non-human primate data. So what we did was, again, we searched PubMed and we gathered all the papers that were using in vitro techniques just from human cells, not from animal cells, focusing on major depressive disorder. We did the same search for in silico and the same search from non-human primate papers. And then we saw how many citations these papers received and by which kind of papers. And also we analyzed the human papers that cited the 3 of them and verified what was the relevance of the citations for the citing paper. Being non-human primates, as I explained to you, very restricted and only used when the benefit is supposed to be higher, one should expect that the non-human primate papers would receive more citations from human medical papers, and its citations to be more relevant for the hypothesis or method of the human medical papers. But once again, this was not what happened. Here you can see the results. And if you look into the total number of citations, you can see that in vitro is the most cited method. It is more cited significantly than non-human primate papers and then in silico papers. Non-human primate papers were also more cited than in silico papers. But if you remember from other studies on the animal models, the majority of citations that the animal model papers received were by subsequent papers on animal models. And here it happened the same. The majority of citations these non-human primate papers received were from subsequent animal papers. So when we look into the citations only by human medical papers, we can see that both in vitro and in silico are more cited than the non-human primate papers. When we look into the citations by human papers, specifically on this disorder, on major depressive disorder, we can see that in vitro is again more cited than the other two approaches. When we look into the relevance, and these results should be appreciated with, should be interpreted with caution because the sample size is small, and it would be actually very interesting to conduct this study in 10 years' time, because in 10 years we will have more data, and it will be interesting to know if the tendencies are the same or if there was some change. But with our sample, what we can see here is that both in silico and in vitro approaches were cited with relevance to the hypothesis or methods 25% of the times, which is better than non-human primate papers. So to conclude, I had a slide here, but it went away. But to conclude, here we can see that not only non-human primate studies do not seem to contribute significantly to the comprehension of this human disorder, but also in silico and in vitro based approaches seem more promising. Then my colleagues and I started wondering, why do we always see this pattern that the animal papers, they are always, the vast majority of the citations these papers receive are always coming from subsequent animal papers. Why is that? Could this be the case that we need many studies, we need animal studies to feed more animal studies before we reach a medical breakthrough? Or is that for another reason? To try to answer this question, we wrote a book chapter that is published in this book, Animal Experimentation Working Towards a Paradigm Change. And our chapter is called, Is Animal-Based Biomedical Research Being Used in its Original Context? And so we looked into, so the hypothesis, if we need a big body of evidence from animals before reaching a breakthrough, then the citations animal papers received should be made mainly by studies focusing on the same disorder. So we took 25 papers randomly from my database of both ADHD and MDD, and we went to verify if the citations these 25 papers received from animal papers and from human papers, if the citations were from the same disorder or from different disorders. And as you can see here, we can see that regarding the, oops, sorry, I can see the full screen because of the Zoom thing here. We can see here that the citations by animal papers are mainly on other subjects and not on the same subject. And this happened in both disorders, in ADHD and in MDD. Regarding the citations, the papers received by human medical papers, there is no difference. And so to answer our questions, do we need animal studies to feed more animal studies before a medical breakthrough? Apparently not. Apparently animal papers seem to be feeding subsequent animal papers, at least in these disorders. I don't know about the others. And so now another question popped out in our minds. Is the current paradigm of biomedical research and drug discovery actually being followed, at least in mental disorder research? Because as you remember from the beginning of my talk, this is the current paradigm, and we have an idea, a drug or a chemical that we test in a simple model, like computer model or in vitro model, then it moves to animal models, then to humans, and then it's proved correct. But apparently, from we have seen so far, the research in human beings is not using that much information of this. And so we were curious to understand if this step was also being fulfilled or not. And so we wrote another paper called "Are in vitro and in silico approach used appropriately for animal-based major depressive disorder research?" And here, what we went to look were from the papers, that the in vitro and in silico papers that described data to acknowledge, to understand major depressive disorder. We went to compare how many citations did these papers received from animal papers. And if the model is the paradigm is being followed, then we expect that these papers here receive more citations from animal model papers than from human medical papers, which should be citing these papers and not these ones, if the paradigm was being followed. But this is not the case, neither for in vitro, neither for in silico. As you can see here, the in vitro papers, they receive more citations from subsequent in vitro papers and from subsequent human medical papers. And the same from in silico. They receive more citations, these papers, from this human medical papers and from the same method. Oops, sorry. But they receive very few citations from animal studies or from the other method, in vitro or in silico. So what we can conclude is that the paradigm is not actually being followed. So from my empirical data, what I can take is that animal studies are not contributing substantially for knowledge of major depressive disorder, at least with rats and non-human primates, and attention deficit hyperactivity disorder. This tendency is also visible in other mental disorders like eating disorders. This is the PhD thesis I told you about in the beginning of my presentation. And it is reasonable to assume that this tendency is present in most, if not all, mental disorders. My results are in agreement with similar studies on other disorders, other non-mental disorders. And other approaches in the in vitro and in silico approaches, especially in vitro in my study, seem more promising. And this has been suggested many times by different authors using different methods. So as you remember from the beginning of my talk, for many years, there was an ethical discussion where people, everyone accepted that animal experimentation was harmful, but it contributed to biomedical progress. And this discussion was if we had the right to use animals in such ways or not. And now we are in the utility discussion, and more data is questioning if animals are in fact useful for biomedical progress. And I think we should move very quickly to a paradigm change in biomedical research. And one way of making this change is the way we propose here, me and my co-authors, in this paper, which is called Ethical and Scientific Pitfalls Concerning Laboratory Research with Non-Human Primates and Possible Solutions. And even though this is focusing on non-human primates, I personally believe that we should be doing this for non-human animals in general. And here what we advocate is that one of the big problems with biomedical research with human models is because we use different ethical frameworks to guide biomedical research with humans and with non-human animals. With human beings, the guidelines are deontological. And the deontology tells us that there are a set of rules and principles that we should follow regardless of the consequences. And this is very clear in all guidelines of biomedical research with human beings. You have to follow the principle of beneficence, of non-maleficence, of autonomy, and of justice. And regarding non-human animals, all the guidelines and legislation are utilitarianists. Utilitarianism tells us that to decide if an action is good or action or ethical, we have to put the costs and benefits of all the involved in a scale and see if the benefits are way more than the costs. And this is what is proposed in the legislation. And I like utilitarianism as a framework, but the problem is that it cannot be applied to this context. And why is that? Because you cannot predict all the benefits and you cannot predict all the harms. So when you judge the harms against the costs and benefits, you are not doing a good job, because you cannot assess the costs and benefits realistically in this research. And so what we propose is that we should use a similar approach with humans and non-human animals, and this will benefit animal welfare and patient care. So my take-home messages is that we need a paradigm change in biomedical research. Animal models should not be assumed to be good models without empirical evidence, because this is a problem. Before these last few decades, where people started questioning, and opposite to what happens with in vitro and in silico, these techniques, before they are being accepted, they have to go through a lot of scrutiny, peer review. They have to prove to be correct. I'm just going to make a small parenthesis to tell you a true story regarding an in vitro method that was developed to test toxicology in the eye. And this was made to replace the DRACE test, which basically is to put the drug or the product directly in the rabbit eye. And this is a very cruel test, and it has also been proposed to be banned. And many years ago, a group of scientists, they were able to reproduce a human eye in vitro, and it could be used to test chemicals. And it was rejected because the correspondence to the rabbit eye was 60% and not 100%. And 100% was the correspondence with the human eye. And so what they did was they created a rabbit eye, and then they could show the regulator that the rabbit eye was a match to the in vitro rabbit eye. And so we should use the human eye, which is 100% correspondent to human eyes. And so this is just for you to understand the major complications that these innovative technologies have to go before they are accepted by the regulations and by the society. But animal models, on the other hand, they are assumed to be good models. It's not mandatory to collect data if they are good models or not. And at least in mental disorders research, they don't seem to be very good models. It should be evident. It should be mandatory to do evidence-based evaluations on the contribution of animal models to biomedical progress in all fields. It should not just be accepted, it should be investigated. And then we can know if there are areas where animal models are in fact useful or not. But we cannot just assume that. We need to investigate that. This is a pyramid on the quality of information. And as you can see here, the less, in the base of the pyramid, the less reliable source of information is background information, expert opinions. And then the top of the pyramid, we have the meta-analysis, which is basically systematic reviews with statistics. We are relying on animal models based on background information, expert opinions, not based on evidence, on the top quality of information. And I think that citation analysis should also be mandatory, because systematic reviews are a very important and interesting tool, and they are mandatory for many fields, and in which you evaluate, you gather the information. For example, if you want to conduct a study on a rat to study major depressive disorder, you are required to gather information on previous studies that were conducted during using rats for major depressive disorder, and you have to see if these studies were well conducted or not, if they are randomized, if they follow good procedures, and if you should expect them to work or not. But when you do this, you leave outside all the other methods, because in your search, you will look for rat major depressive disorder, so you will not see what is being done in vitro and in silico. So this does not prevent studies that could be conducted without the animal models. And also you don't see retrospectively which was the impact. And citation analysis can answer that question, and I think they should be more widely used. I can't see what's is in the bottom because of but I think it's the sentence I just, yeah, it's the sentence I just read. Oops. Sorry, I don't know what's happening. I'm trying to get to the next slide and it's not working. Okay, so in the beginning of my talk, the name of my talk is Animal Use in Mental Disorders Research, a Necessary Evil. And I would like to end with a bit of a provocative question, which is, if we look into the expression necessary evil and we cut necessary out, which is apparently the case in many of these disorders, then we just have the word evil. And this is just to open the debate. Before that, I would like to thank the co-authors of all of my studies and also Animal Free Research Foundation, who sponsored my tuition fees of my PhD, and the Johns Hopkins University Center for Alternatives of Animal Testing, which gave me an award last year, and of course, Centro de Filosofia das Ciências da Universidade de Lisboa, which is my center at university, and that invited me to be here today, particularly João and Silvia, who are the ones that sent the invitation.